Cue camera. All right. Today, as I'm going to let you do the copies of the sideboards. You can do that. But because I think these introductions are important, what I'll do for today's lecture is I will mount the introduction from last semester, which is pretty much identical to this one as a separate link. And I'll put that under the link for the lecture that we'll have today. And then you can go back and check on that and see what was said to introduce it all. And there wouldn't be much difference between what I would say here today and what I said last November. So the only real difference is last November there's a welcome back from Thanksgiving break. But what we're going to do now today is we're going to start into this idea of the different kinds of digestion. And we're going to start here with bulk phase digestion. And this occurs in the center of the intestinal lumen. So I'll briefly show what I mean by that. Whereas if this is meant to be the intestine, remember that the mucosa of the intestine is thrown into folds. But even after it's thrown into these folds, there's a sizable region in here, which I'm going to label bulk phase. This is going to be almost all the volume of the small intestine. And this bulk phase is going to be churned by smooth muscle contraction. So this is a lot like a washing machine out here. This fluid is just moving back and forth and back and forth. And what we're going to see is that in this bulk phase, we have three main actors. The first one is going to be pancreatic amylase. The second is going to be lipase. And the third is going to be trypsin and chymotrypsin from trypsinogen and chymotrypsinogen. Well, let's take it from the bottom up. These enzymes, trypsinogen and chymotrypsinogen, you'll recognize as inactive precursors. They will be secreted by the pancreatic acinar cells in response to CCK. Remember, the CCK is being released by the eye cells in the mucosa of the small intestine as a proper hormone. The cholecystokinin goes from the mucosa of the small intestine and binds to the acinar cells in the exocrine pancreas. That then leads to the release of trypsinogen and chymotrypsinogen. But then in the small intestine, the trypsinogen and chymotrypsinogen is converted, to active proteolytic enzymes. Now, the function of this, of course, is to convert proteins to peptides. Now, this distinction between proteins and peptides is simply a matter of size. When we talk about a protein, we typically figure that there are maybe several hundred or a few thousand amino acids. Whereas in peptides, these are usually shorter lengths, maybe 20, 30, or 50 amino acids. So we're basically taking the great big proteins and converting them down to the small peptides. Similarly, pancreatic amylase acts on starch and converts starch into maltose. And again, remember that starch is this great long polymer of glucoses, and that maltose is constructed from a glucose plus glucose. Now, the middle player here, lipase. Remember, lipase is going to be working on a hydrophobic molecule. Lipase converts triglycerides into two fatty acid chains and one monoglyceride. Now, I'll introduce it here, and we're going to come back to it. You'll recognize this becomes a problem when you realize that the lipase is an aqueous enzyme. It's an enzyme that's working in water. And yet it has to attack this blob of fat and convert the blob of fat into monoglycerides and fatty acids. So you have this gigantic beach ball of fat that can only be digested by something in the water layer, which can only be digested at the surface. So one of the ideas we're going to move through here today is that lipase converts triglycerides into two fatty acids and one monoglyceride after emulsification. With bile salts from the liver. I'll put this in here via the gallbladder. This is going to be the initial phase of breaking down the the initial phase of breaking down triglycerides into their component parts. But basically what we're going to do is we're going to convert them to monoglycerides and fatty acids and then allow them to go into the enterocytes. And in the enterocytes, we're going to then rebuild those lipids in the smooth endoplasmic reticulum and in the Golgi apparatus, complex them with proteins, and then shove them to the outside as chylomicrons, and we'll talk about that toward the end. But all three of these are occurring here in the bulk phase, where things are squishing back and forth. Let's take a minute here and just look at the slide, because I think it will emphasize a couple of things. First is this image of the small intestine, where we have this open tube, and the open tube is lined with these folds, and then the folds are constructed from the constructed from the villi. That's a villus. We talked about that last time. A little bit later here, we're going to delve down and talk about the structure of individual enterocytes here. But what I wanted to show you right now is this structure, to remind you of a couple of things. First, that this yellow structure is intended to be the pancreas. And what it's trying to show is that there are tubes in the pancreas, and these tubes are part of the exocrine pancreas. And if we were to blow this up, we would find that these tubes are lined with epithelial cells. Some of those epithelial cells are part of the duct epithelium, and they will be secreting bicarbonate. Others will be part of the acinar cells. Those will be the bulbous-like structures on the endocrine pancreas, and they will respond to cholecystokinin and release the enzymes you just put on the board. But the fate of those enzymes now is to move down into the small intestine. By the end of the day today, we need to get to the gallbladder and the secretion of bile salts and bile acids into the small stomach, into the small intestine, so that we can now begin to emulsify the lipids. But if you hold on to this image and recognize that we're going to be secreting bicarbonate, and from today's lecture, the pancreatic amylase, lipase, and proteases down into the small intestine. It's now in the small intestine where these molecules, these enzymes, will have their action. And these guys are going to have their action in the middle of the tube, where things are being stirred back and forth. Because what we're going to do now is we're now going to try to imagine, rather than being in this stormy middle of the intestine, we're now going to go right down to the edge of the wall of the intestine, within about 30 micrometers of the wall of the intestine. So this will be within about 30 micrometers of the villi. So we're going to bring this fluid right down to the surface of the small intestine. Now, for those of you who have had some fluid mechanics, you'll recognize this immediately. But for the most of the rest of you, you'll have to take it more or less on faith, that there is about, a 30 to at most 40 micrometer thick layer of unstirred fluid over the enterocytes. And it's fair to say over the enterocytes of the villi. Now remember that when we talked about the villi, we drew them in such a way that we described a continuous layer of enterocytes, the epithelial cells of the small intestine. So each of these hashes is meant to represent an enterocyte, and there'll be thousands and thousands and thousands of these. And they were oriented such that they had a basilar surface sitting on a basal lamina, as with all epithelia. And then beyond the basilar surface, there was a blood vessel. I'll put this in, I may as well put it in red. The blood vessel was part of the hepatic portal vein. And then residing right next door to that blood vessel's part of the hepatic portal vein was another tube. And this tube is a lymphatic capillary. In the intestine, these lymphatic capillaries have a name. They're called lacteals. So that's the overall structure of the villus. So this is the villus. Now what I'm going to do on the next part of the board is we're going to take a series of these, a small stretch of these individual enterocytes, and show them with respect to the unstirred layer and how they're organized with respect to one another. So each of these cells is no more than about 10 micrometers thick. And if we look at these cells, so this is an enterocyte. And the enterocytes, of course, are all stacked side by side. And to orient you here, down here in this part is the basal lamina. And then between the cells are these tight junctions. We've seen this any number of times when we talked about epithelia. So this is meant to be the tight junction. Down here, and I'll put these in red. Down here are going to be endothelial cells. This is meant to be an endothelial cell of the hepatic portal vein. It's actually a capillary of the hepatic portal vein. But now what I want to do next is I want to look at the apical surface of these epithelial cells. And if we look at the apical surface of these epithelial cells, they're thrown into these structures that are finger-like projections. Each one of these cells has these finger-like projections on their apical surface. And these finger-like projections have a name. They're called microvilli. Now again, the villus is a multicellular structure. So it has thousands of these individual enterocytes. But each individual enterocyte on its apical surface has its plasma membrane thrown into these folds called microvilli. It's all about increasing the surface area. So what we do is we increase the surface area for the uptake of small molecules here by throwing that apical membrane into folds. Now in orange out here, I'm going to put in a box. And this box is the (20 to 30 micrometer, well, let's say 30 to 40, fine, 30 to 40 micrometer wide, or thick, unstirred layer. So we've seen this before. Every time we get closer and closer to the wall with a fluid, there's a thin layer that doesn't move hardly at all. And that's true in the small intestine as well. So this is a layer of fluid, mighty thin, but it's not being churned back and forth. If we go up this way into the lumen of the small intestine, we're into the area that's being churned back and forth. That's the region of bulk digestion. But this is the unstirred layer. And here in the unstirred layer, molecules move by diffusion. So let's take one of the molecules we talked about that's generated by the pancreatic amylase. We said that's going to make maltose. So there will be maltose out here. And the maltose will be being squished around in that stirred layer, but eventually it will find its way down here into the unstirred layer, move slower and slower until it finally enters the unstirred layer. And remember, this molecule of maltose is made of glucose plus glucose. Once this molecule gets into the unstirred layer, it now can simply move by its random walk of diffusion. It's not being pushed by anything. It's just movement by thermal agitation. So it's just kind of wandering around in here. But then we'll find that on the surface of these cells, as transmembrane proteins, is an old friend. This is the disaccharidase. maltase. And what the maltase does, as the name implies, is it converts maltose to 2 molecules of glucose. So again, we've taken, in the bulk phase, we've taken these gigantic molecules of starch, and we've broken those gigantic molecules of starch down into much smaller components, the 2-hexose sugar components of maltose. Now, the bulk phase has no way of dealing with the maltose beyond that. The only time we can deal with the maltose now is to use this so-called brush border enzyme called maltase. I should say at this point that taken together, the microvilli form the brush border. Many of these names are left over from simple anatomical descriptions. But we now know the microvilli are plasma membrane extensions of the enterocytes. And one of the major players in here is the disaccharide maltase. It converts maltose to glucose, and now right nearby in another cell here can be a molecule which we've seen before called a sodium-driven cotransporter. So now in this case, the generation of glucose from the maltose, these glucose molecules are now randomly walking about, and they'll strike, on occasion, one of these sodium-driven cotransporters. And if a sodium ion is attached to the sodium-driven cotransporter when it binds to glucose, then into the cell comes glucose plus sodium. Where have we seen this before? Better know by Wednesday. This is in the proximal convoluted tubule. So this is a sodium-driven cotransporter. And similarly, down here, there is this gigantic protein called GLUT2. GLUT2 is a facilitated transporter, just as we saw in the kidney. So we can think of this as just a sort of modified structure of the proximal convoluted tubule. So the glucose comes in, moves across the basolateral membrane, out into the extracellular space, and then into the hepatic portal system. Well, we can do the same thing here with proteins. We said that in the bulk phase, trypsin and chymotrypsin are now converting these great big molecules of proteins into peptides. So now we'll have a string of these peptides, and each one of these circles is meant to be an amino acid. And maybe there are 30 or 40 or 50 of them. But they're fairly small molecules. And of course, they have an amino terminus and a carboxy terminus. And on these brush borders, along the microvilli, will be transmembrane proteins here. These transmembrane proteins are going to be amino and carboxy peptidases. So what they're going to be doing is they're going to be clipping off either an amino acid at the carboxy end or at the amino end. So when these peptides, 20, 30, 40 amino acids long, come close enough to the transmembrane protein of the amino and carboxy peptidases, those guys will clip loose an individual amino acid. Then again, just as we saw before, use green. Just as we saw before, there are going to be these transmembrane proteins in the form of a sodium-driven amino acid transporter. The genes that we saw in the proximal convoluted tubule that were expressed to transport amino acids into the cells in the proximal convoluted tubule are the same genes expressed here. And what we're going to do now is we're going to bind an amino acid that's been clipped loose of the peptide along with an ion of sodium, and again, transport across the apical epithelium an amino acid and sodium. And as before, down here, there is this amino acid facilitated transporter. Same story as the kidney. We bring the amino acids in. There's this big protein at the bottom. The amino acid then diffuses out across the basolateral membrane and enters the hepatic portal system. So in this way, we have now converted the great big molecules found in the bulk phase down into smaller pieces, and then moved those molecules across the epithelium into the blood vascular system. And those will then be headed to the liver. Before we get to the liver, however, there's a couple more stories I want to tell you. There's another molecule out here, and I'm running out of colors, but put it in brown. There's another molecule out here as a transmembrane protein, one of these brush border enzymes, known as sucrase. So when we eat sucrose, when we eat sugar, we take in a fair amount of sucrose. And sucrose, of course, is composed of glucose plus fructose. Well, there really isn't any bulk phase digestion of this molecule. This is all going to occur on the brush border. And when these molecules of sucrose get down here, they'll be snagged by the sucrase. And they'll be converted into a molecule of glucose and a molecule of fructose. The glucose then can be transported across the membrane by the sodium-driven glucose transporter. Now, for those in biochemistry, it turns out that fructose has a special transporter. It's called GLUT5. It's a different transporter. It's the same on the apical surfaces of the basolateral surface, but let's forget it there. We'll just leave it as the fact that there's another means of getting fructose in, but let's not worry about it. Let's really stick to the idea of the sodium-driven co-transport of glucose. And you remember there was another character that we mentioned when we described the disaccharides. And the disaccharide that we mentioned was, running out of room, lactose. This is milk sugar. It's unique to mammals. And lactose is composed of a molecule of glucose plus a molecule of galactose. If you actually look at the structure of glucose and galactose, they're remarkably similar. It's just on the number 4 carbon, the position of the hydroxyls switched from being up to being down. So don't worry about it. These are really similar. So in all mammals, what we find is, as a transmembrane protein here, a molecule of lactase. And the lactase converts lactose into a molecule of glucose plus a molecule of galactose. Now that's all out here in the unstirred layer. So again, we're going to need some sodium-driven cotransporters. So again, we have this sodium-driven glucose transporter. So it can then bind a molecule of glucose and a sodium ion and do the same thing. Bring the glucose in. Bring the sodium in. There will be a facilitated transporter on this side, GLUT2. And the glucose now goes into the blood vascular compartment in the hepatic portal system. Now, you'll remember that milk is really unique to mammals. After all, the mammary gland is from which the term mammals is derived. And so this means that all mammals are raised for the first period of their lives on lactose. It's true for humans as well. But there's an interesting thing that happened about 7,000 years ago. About 7,000 years ago, looks like first place it occurred was someplace in Central Europe, someplace around present-day Ukraine. There were a group of people who started to domesticate these great cow-like animals. They were wild animals called aurochs. They're extinct now, but they stuck around until about 1600. And the aurochs then were domesticated, and for a long time, people simply kept them sort of in the backyard. And then they would now and again go out and kill an aurochs because it was easier than going and hunting them. But then someone got a bright idea. They said, well, the cow feeds the calf on milk. Maybe we can manage to use the milk too. So they started drinking milk. But it wasn't very useful to them. The reason it wasn't useful to them is because at the time of weaning, there's a gene that says, stop making lactase. You don't need it anymore. You're done drinking milk. Get rid of the lactase gene. And so in mammals, there's an upstream regulator that says, turn off the lactase. And this happens in almost all mammals. It appears that in the neighborhood of about 7,000 years ago, there was a single individual in this group in Central Europe that had a mutation in the upstream regulator. And it's a single site mutation. I think it's a G to an A, single site. And when that happens, the upstream regulator doesn't tell the lactase to turn off. It makes a mistake and leaves the lactase on. That means those individuals who have a mutation in the upstream regulator can continue to have lactase embedded in their brush border membranes. And they can continue to digest lactase or lactose. So if you look at the populations of Northern Europe, you find that those populations that are up and around Sweden, Finland, Northern Germany, England, all of that, 95% of the population there can digest lactose. If you begin moving farther and farther south into Europe, the presence of the lactase gene gets to be less and less. And you can follow this culturally. In those places where you have raw milk products, pretty much Northern Europe. As you move farther and farther south, you begin to find fermented milk products, because now you're converting the lactose into simpler sugars by the fermentation process. Now, if you go to parts of East Asia where dairy culture never grew up, like 95% of the population is lactose intolerant, meaning they can't digest lactose. Now, this would be just a simple story, and we couldn't really bank on it very much. But there have been several cases where a dairy culture arrives. One case was around Pakistan. It looks like that was done with sheep and goats. There was another case in camels that cropped up around Saudi Arabia. There's another case in Eastern Africa that appears to have cropped up only about 3,000 or 4,000 years ago. And each of those cases, within 1,000 years or so of the rise of taking care of animals for milk, dairy industry, there comes this gene, this repressor gene gets turned off. In each of those cases, it's a different gene mutation. It's a different single site mutation, but in each case, it turns off the repressor so that the adult generates lactose. And in most cases, those are dominants. It's a gain of function mutation. So as long as you turn off one of the genes, one of the two repressor genes, and don't make it repress the generation of lactose or lactase, then you can continue to make that enzyme and digest milk. Yes?
So when lactose is converted to glucose into lactose, did both glucose and lactose go into the cell?
So the question here is, how does the lactose get from the outside of the cell across? And it turns out that the sodium-driven cotransporter for glucose works every bit as well for lactose as glucose. It doesn't matter about that change in the hydroxyl. So any questions up to there? Because now what we're going to do is we're going to deliver this stuff to the liver. We're going to carry the blood to the liver. We've managed to get these small molecules into the blood supply in the hepatic portal vein. Now we want to take it to the liver, and that's what I'm going to do in the upper panel. And I'm going to draw this. I'm going to draw this as something known as a liver lobule. Now, the liver lobule is a subunit of the liver, and it's a multicellular structure. It's about 2 to 3 millimeters in diameter. It's a sizable structure, but it's composed of hepatocytes, and it has a particular blood supply that I want to tell you about. Now, when we look at a liver lobule, when you look at it in the microscope, what one is struck by is that it has this unusual hexagonal shape. So this is meant to be a liver lobule. And at each of these vertices, we see 2 blood vessels. One is a branch of the hepatic artery, and it's carrying oxygen-rich blood. And something like, about a third to maybe as much as a half of blood to the liver is hepatic artery blood. But meanwhile, right at these vertices, I'm going to put this in blue, there's this structure, which is a branch of the hepatic portal vein. And at each one of these points, we have the same pattern. And sitting next door will be in red an extension of the hepatic artery. Now, these will empty into a structure here called the central vein. And this will empty into the hepatic vein. But if we trace these back, we see that they are effectively capillaries. They're called sinusoids. And they are lined with endothelium. And each one of these now is effectively a tube that drains the blood into the central vein. one up here too. So now, at this point, we're mixing together the oxygenated blood from the hepatic artery with the deoxygenated but nutrient-rich blood of the hepatic portal vein. So we're mixing these two together. So coming in blue here is O2 poor blood that is nutrient rich. Sitting right next door to the endothelial cells are these brown squares. Now these brown squares are meant to represent, hepatocytes. If we come over here and describe the endothelial cells here, this is an endothelium, this is the sinusoid, sitting right next door to the basilar surface of those endothelial cells are hepatocytes. And what the hepatocytes are doing is they're taking up glucose and they're taking up amino acids, which we had deposited in the hepatic portal blood. That's the story from above. Now, once these amino acids come into the hepatocytes, they're reconstructed into proteins. And glucose is reconstructed into glycogen. Glycogen looks like amylopectin, looks like starch. It basically is animal starch. It's a highly branched structure of glucose. So basically, we're simply rebuilding the glucose molecules back into a polymer. And we'll store it as a polymer here in the liver. And when we need more glucose, we'll mobilize the glucose from the glycogen and put it back into the blood. But for our purposes now, what we're doing is using these hepatocytes to store the individual molecules we broke down and transported across the intestinal epithelium. But there's another set of cells in here I need to tell you about, alongside these hepatocytes. And these form a kind of tube. They call this a canaliculus. And the canaliculus takes up a molecule called bile that is synthesized by hepatocytes. Now, a word on bile. We talked about bilirubin and biliverdin being a waste product, but bile is composed of a molecule of cholesterol linked to glycine or taurine. Now, the point of that is that the cholesterol is hydrophobic. So if we looked at bile, if we actually look at a molecule of bile, it has this cholesterol structure. And out here, it's linked, I think it's on this number 5 carbon ring. There's another one out here. A, B, C. I'm sorry, this is a 6-membered ring. This is a 5-membered ring. And out here, you put a taurine or glycine. Now, the absolute structure, this is not important. This is charged. This is uncharged. That means we have an amphipathic molecule we have constructed a detergent. So the long and the short of that is the hepatocytes are synthesizing a detergent, putting it in the canaliculi, and the canaliculi empty into the gallbladder. The gallbladder is just a storage vat. It's a smooth muscle-lined storage vat. And you remember the term cholecystokinin, bile sac move. When you secrete cholecystokinin, it not only binds to the acinar cells, but also binds to the smooth muscle in the gallbladder. And you remember from the first slide I showed you, there was that sac. It was in green. And when the smooth muscle contracts, it squirts this bile into the small intestine. Now, the last couple of minutes, let me tell you what the bile does. I'll put this in the backboard. Bile is a detergent that emulsifies fat. Basically, it takes these great big globs of fat, which doesn't interact easily with water, and by interacting with the globs of fat, it converts them down to tiny BBs. You can imagine taking a beach ball and converting it into these tiny BBs. So now we increase the surface area to volume ratio. That's the role of bile. It increases the surface area to volume ratio so that we can now attack these with lipase. And when we attack these BBs with lipase, what we end up with is in the BBs, we end up with 2 molecules of fatty acid and a single molecule of monoglyceride. These are effectively my cells. A minute. When the micelles approach the microvilli, the micelles, the components of the micelles, because they're lipid soluble, easily go right across the lipid membrane. And so now we have monoglyceride and fatty acids inside the cell. Now, oddly enough, inside the cell, these monoglycerides and fatty acids in the ER are reconstructed into triglyceride. And then in the Golgi apparatus, the triglyceride is complexed with protein, so that now from the basolateral surface, we can release these tiny particles, about 1 to 10 micrometers in diameter, that are called chylomicrons. The chylomicrons now, those chylomicrons go into the lymph and then are carried in the lymphatic system back to the subclavian vein and dumped into the lipid. And then adipocytes reconstruct the whole thing all over again. It's just a bizarre Rube Goldberg device to deal with lipid. With that, I'll let you go. That's my last lecture, and thanks.